Hello, this is Dr. James Camp from Lee College in Baytown, Texas, and this is my lecture on the specific immune system. We talked previously about the two first lines of defense, that is, barrier methods that keep an infection from happening, and non-specific responses that try to prevent an infection from taking hold. What we're getting into today is the third and most powerful line of defense, the antigen-specific responses, the B cells and T cells that track down an established infection and destroy it. So the first question that may come to mind when we say a specific immune response is, well, specific to what? What we're trying to do is, what your immune system is trying to do is to identify a specific pathogen. So let's say we have a bacteria here. That bacterium is surrounded by a plasma membrane, but that plasma membrane is exactly the same, more or less, as the plasma membrane of your own cells. There's no way to be specific about identifying that bacterium so far. But we recognize that bacteria, just like us, have proteins that they post on their cell membranes. So maybe this bacterium has a red box shaped protein on its cell membrane, and also it has a green triangle shaped protein on its cell membrane. These proteins are now able to play the role of antigens. An antigen is something that produces an antigen response, anti against, generate, give rise to. So it generates a response against it from your immune system, from your specific immune system, to be specific. What if we also had a virus over here? And that virus had these little purple circles coming off of it that helped it infect your cells. And also maybe it had these orange squiggle proteins that were part of its basic capsule makeup. In that case, assuming your body doesn't make its own purple circle proteins or red squiggle or orange squiggle proteins, those also can play the role of antigens. So both invading microorganisms and invading viruses have the ability to be covered with these antigens that your body, that your specific immune system can respond to. What are some characteristics of, what makes a good antigen? The answer is three things, okay? One, it has to be chemically complex. Proteins with their 21 possible amino acids make very good antigens. Some other things that make good antigens are complex carbohydrates, such as those found on glycolipids or glycoproteins, and also certain other artificial molecules that we make. There's a reason that we don't make medical implants out of wood. Those have chemically complex surfaces that would trigger an immune response. What we tend to do instead is make chemical biomedical implants out of chemically simple materials like titanium metal or polyethylene plastic. Two, the good antigen should be different from any molecules that you make on your own cells or in your own body. In immunology, we refer to these as self-antigens, antigens that describe what your self looks like to your own body. And then 3, they should be relatively abundant. If we had a little, a single lime green oval here on the back of this bacterium, that would not make a good antigen, because you would have to precisely target that bacterium in that one place to find it. Whereas the red squares and the green triangles are all over, they're relatively abundant, they make very good antigens. Okay, well, now that we know what an antigen is, you may also be wondering, well, how does our immune system recognize it? And the answer is with 2 things. One is antibodies and the other is cell receptors. An antibody is this funny looking Y-shaped molecule consisting of 2 angled long chains and 2 bar-shaped short chains. At the tip of each chain is a region called a variable region that is randomly generated in each different line of B cells or T cells. Well, B cells make antibodies, so it would be B cells. This one looks like it recognizes our green triangle proteins from the previous page. Essentially, this is what's called an antigen binding site, and it acts as basically a molecular recognizer. Okay, and so that molecular recognizer is going to stick to only one specific antigen. Okay, because there are millions of possible antigens out in the world. When your immune system is beginning to develop as a fetus, you generate millions of random combinations of possible antigen binding sites on millions of B cells that each create their own antibodies. How does your body know which ones to produce? Once you've generated a million possible combinations, it waits and it sees if that particular B cell finds its particular antigen trying to invade the body, and if it does, then it starts churning. It activates that B cell and it starts churning out antibodies to track down and fight that disease. Well, then how does the B cell know that its particular antigen is there in the body? Well, B cells and T cells, or B lymphocytes and T lymphocytes, as the more proper terminology would be, have something very similar to antibodies on their cell surface. These are called receptors, and so the B cell that would make this particular antibody is going to have a B cell receptor that looks exactly like the binding site, the antigen binding site for the antibody it's going to produce. That is a B cell receptor, or BCR for short. The T cell over here might instead respond to our red boxes, okay? Or maybe, better example, because T cells are more antiviral, maybe it will respond to our purple circles, okay, from the virus on the previous page. So that T cell has a T cell receptor, or TCR for short, that would recognize its particular antigen, the particular antigen that it would respond against. Okay, so I mentioned that we generate millions of possible B cells and T cells before we're even born, but that most of those stay dormant most of our lives. They stay inactive, and they have to be activated specifically in order for an immune response to take place. How does that activation happen? Well, the first thing that has to happen is the antigen has to be recognized, and it has to be presented to the specific immune cells to get this party started, so to speak. So let's say we start with a pathogen of some sort. Can be a bacteria or a virus. And let's say it's got little green circle proteins coating it. The first step in our reaction, of course, is that the pathogen would infect our body. But that's really kind of step 0. Step 1 of the process then is some sort of a phagocyte, maybe a macrophage or something called a dendritic cell that lives in your lymph nodes, would gobble up this pathogen, and that macrophage recognizes that this pathogen is something that needs an immune response to it, that it's not going to be able to gobble up all of these pathogens on its own. So the macrophage or dendritic cell or other cell immediately converts itself into an antigen presenting cell, or APC for short. And the antigen presenting cell uses something called the major histocompatibility complex, an ability that all cells have to display antigens for inspection by the immune system. But instead of presenting its own antigens, it presents the antigens from the pathogen that it just swallowed up. So here it is, coated with this green circle protein, ready for the immune system to see it and become active. Well, along comes an inactive T helper cell, or TH for short. These are helper cells because they don't launch a full-blown immune response themselves. They just help to start one. Okay, and it's got its little T cell receptors on its surface with green circle recognizers. And voila, there's a binding event that occurs between the T helper and the antigen presenting cell. And so what happens next is that the inactive T helper activates, and it also clones itself. It divides multiple times to produce a clone, a group of active T helpers ready to go off and help the other cells of the immune system become active. What are those other cells of the immune system that need to become active? Well, we've mentioned B cells and T cells. You know that B cells are the ones that produce antibodies. That's technically not quite true. The B cells have to differentiate into another type of cell called a plasma cell. So here we've got an inactive B cell sitting here minding its own business. It's got its little B cell receptors, and these happen to also be green circle recognizers. Well, the first thing it does is it runs into our pathogen with its green circles all over it. And there's a binding event there. Some sort of recognition happens that the inactive B cell sees that its pathogen is now trying to infect the body. Now is the time to launch a response. At the same time, our T helper cell from the previous page comes in and it sends a signal to the B cell called interleukin-2, or IL-2 for short. That's inter between leukos, white, so between white blood cells, interleukin. Interleukin-2 encourages the inactive B cell to take this pathogen seriously and become active. So the inactive B cell does pretty much what you would expect. It becomes active, and just like the T cell before, it clones itself into a whole bunch of active B cells. Most of them are now what we call plasma cells. And those plasma cells are busy secreting antibody with those green circle recognizers on them. And so they will all produce lots and lots of that antibody. It will circulate through your blood and your lymph, and everywhere that they encounter one of these pathogens, they will stick to the pathogen and label it for immediate removal or destruction or something like that. The antibody itself does not usually kill the pathogen, but it makes it impossible for the pathogen to keep infecting the body, and it causes the body's natural defense mechanisms to more specifically attack and destroy that pathogen. At the same time, a small number of these activated cells become what we call memory B cells. And those memory B cells will linger around in your lymph nodes and your tonsils and other immune surveillance sites, waiting for this particular pathogen to come back a second time. And when it does, they're ready to immediately launch a full-blown reaction. They don't have to go through this whole activation process again, and that makes your second exposure, or third or fourth, to a particular pathogen have a much faster, much stronger, much more effective immune response. Okay, so we know that B cells make antibodies, and those antibodies will take care of the free pathogen floating around your body. But what about your body's own cells that may have become infected and are therefore harboring, or in fact even manufacturing, more of the pathogen itself. Okay? They've become pathogen factories. We need to get rid of those. In most cases, there's no way to heal that infected cell. It just needs to be destroyed. So the worker we have to do that is a cytotoxic or killer T cell, or TC for short. So we've got an inactive killer T cell over here with that same, and it's once again got its T cell receptors on with those same green circle recognizers. And it once again has to have the antigen presented to it, like its T cell buddy, the helper T cell. But once there is a recognition reaction here that the T cell sees that its antigen is being presented to it, once again it gets a signal from our T helper cell from the previous page, again is going to send that interleukin-2 signal over to the inactive T cell to make it active, make it take this seriously. The inactive T cell is going to, you guessed it, clone itself into lots of little active killer T cells. And those T cells are going to attack the infected cell just like the natural killers we talked about in the non-specific immune response. They're going to attack it with chemicals that will cause that infected cell to go through apoptosis, to commit cellular suicide, and in so doing, they destroy that particular pathogen factory and keep the body from getting more and more infected. At the same time, a small number of these cells become memory T cells that again hang around in your lymph nodes until the antigen is once again found in your body and they need to come back out and do their job all over again. And that's the system for activating our specific immune responses. Remember, we have millions of possible T cells and B cells that remain inactive until their antigen is seen, and we need this two-part activation system to specifically activate only the ones that will be useful, that will attack an actual threat to our body and not go haywire and attack something they shouldn't. Okay, two last things I want to talk about. One is the immunization effect. You may have wondered why it is that we bother vaccinating people. Wouldn't they have the same reaction when they finally get the real flu that they would have from seeing a flu vaccine? Well, the idea is that the primary response, the first time you ever encounter a pathogen, it has a short delay period, a short incubation period. For many viruses, it's about 7 days. And then the pathogen starts shooting up, starts really infecting your body. Well, it takes about that same 7 days for your IgG, your blood antibody response, to even get going in the first place, for all of those induction activation steps that we just talked about to happen. And so the pathogen has a serious head start on the immune response to it. But because of those memory cells that we create, the second time the pathogen is found in your body, it has the same incubation time, and then it starts to work its way up. But as soon as that pathogen shows up in your lymph nodes, your tonsils, your spleen, wherever, the blood antibody response starts to absolutely shoot up through the roof. It gets to be, it's not only much, much faster than before, 4 days as opposed to, 15, but it's also easily 10 times stronger than the initial response. And so this time, the poor little pathogen is way overdone by the immune system, doesn't stand a chance to really even establish an infection in the first place, and your pathogen is destroyed and goes away. So the theory behind immunization or vaccination is that if you can get this primary response taken care of with an artificial or a killed or attenuated virus or bacterium, then when the real virus or bacterium, the one that actually could kill you, gets into your system, you have this secondary response, and your immune system just clobbers that virus or bacterium. And that's why we immunize our children. That's why we even immunize adults, is so that when you finally get exposed to one of these deadly, or sometimes not deadly, but just lifelong infections, your body can clear the infection before it even gets established, and you don't ever have to worry about it. The second is that just from a clinical standpoint, there are two ways to gain immunity. Passive immunity is where antibodies are provided passively to a person. For example, through breast milk, babies get their mother's antibodies transferred to them during breastfeeding, or through injection. Sometimes we have anti-sera available to give a patient, perhaps against a snake bite venom, or against a particular virus that we don't have a vaccine for, but we might have antibodies available for it. They did some of that therapy with the latest Ebola outbreak in Africa. They took antibodies from people who had survived Ebola and gave them to the people who had just been infected to see if it would clear the infection, and sometimes it worked. Active immunity is where antibodies are produced actively by a person's own immune cells after exposure to the antigen. That could be simply by being infected by the virus or bacteria, or it could be through a vaccine, an immunization. The thing that's most important to note here is that with passive immunity, there is no development of memory cells, so there is no long-term protection. With active immunity, there is development of memory cells, so that produces lifelong, or at least long-term, immunity. And so there are 4 ways to gain immunity. Natural passive, antibodies from your mother, for example. Natural active, you contract the disease and you eventually produce the antibodies. Artificial passive, you get an injection that's been prepared at the clinic. You don't gain any lifelong immunity from this, but it does help you get through that one episode. And finally, artificial active, which is what we kind of strive for in the medical world, is where a person receives a vaccine, an inactive pathogen, and learns to produce antibodies against it, stores memory cells, and therefore has a lifelong or at least long-term immunity against that pathogen. All right, that concludes my presentation about the active, the specific immune system. Hope you enjoyed it, and I'll see you in class.